So welcome to the second part of the chapter 14, and these are the receptor tyrosine kinases. So as the name shows, these are actually the amino acid tyrosine. It is a type of amino acids involved in this signaling, and we will see how this tyrosine is involved and why they are named as receptor tyrosine kinases. So they are different from the G protein coupled receptors in a sense that the G protein coupled receptors most of the time they are relevant with the physiological processes. So the signaling of the physiological processes is mostly controlled by the G protein coupled receptors. But the whenever there is a growth, right, whenever there is a kind of cell division involved, then these receptors are very important because these are somehow more tightly regulated when compared with the G protein coupled receptors. Why? Because the cell division is a very crucial process. It is a very critical process. If there is something wrong with this process, it will lead to cancer. So that is also one of the reasons that why receptor tyrosine kinases they are they are highly expressive in all types of cancers. Okay, these are some of the examples. You can see the growth factors: epidermal growth factors, transforming growth factors, platelet derived growth factors, interleukins, CSF1. These are the growth factors. They are going to attach with their target cells, and then they will make a complex with their tyrosine kinase receptors, and these complex will lead to different responses, and most of the time these responses are the growth responses. Yes, so receptor tyrosine kinases they are going to initiate a signal transduction cascade that will involve RAS and MAP kinase, right? RAS is a protein that is present in our cells, and it is inactive form when it is bound with the GDP, when it is activated when it is bound with the GTP. So we will see that again that GTP GDP complex is involved here, and MAP it is an enzyme, and whenever there is an enzyme involved, then the name will be kinase. Whenever there is kinetics are involved or the energy is involved, so that enzyme will always be termed as kinase enzyme. Okay, so we will see what will happen to the pathway. Here you see the RAS, RAF, MAC, ERK pathway, and there's so many names, right? And mitogen activated protein kinase cascade, here you can see, and then these 4 MAP kinase cascade in humans. We have then RAS molecule. RAS is a protein, RAF again is a protein, MEC it is a protein, right? MAP kinase, ERK kinase. So this pathway is involved in the growth of the cells. We will see that how that growth factors are involved here. So the most important thing is the RAS protein. RAS it is rat sarcoma, right? Sarcoma means the cancer of the skin. So this was first discovered in the rats, so that's why they are known as the rat sarcoma. And RAF is acronym of the rapidly accelerated fibrosarcoma growth factor receptors, and they are bound with the protein 2. Okay, so these are some of the names of the same process. Okay, so don't confuse it that these are some different processes. These are some of the abbreviations, but the pathway is the same: the RAS pathway or the MEC pathway, MEC-ERK pathway, RAF pathway. Okay, so this is very interesting slide. Here you see the tyrosine residues. Where they are present? They are present in the cytosolicitals. Here you see here on the and look at the arrow here, the outside the cell, and this is the ligand. Ligand is going to bind here, and then there will be structural changes in the receptors. So the receptor tyrosine kinases they always work in pairs. So this is the main difference between the G protein coupled receptors and the receptor tyrosine kinases, that they are going to work in twins, in pairs, in couples. So why? Because they are more tightly regulated. So when there is a signal like the epidermal growth factor ligand is going to bound to the receptor, then the tyrosine residues will autophosphorylate. This tyrosine residue will phosphorylate this tyrosine residue, and this one is going to phosphorylate this one. So this is known as the autophosphorylation. Again, this is an arrangement by the nature for tight regulation, so that there should be no mistake at all. So here you see the first step is that the epidermal growth factor is going to bind with the receptor, and after that you see these are GDP attached with the RAS protein, and RAS protein is inactivated, right, when the GDP is attached. So these are also the proteins. As I told you in the previous lecture, that these are ready-made proteins. They are always present in the cell, and they are present in inactive form. They are not present in an they are not made from the step 1. They are already present. They are just waiting for some signal, and signal is here. When the signal reached to the target location of the receptor undergo changes, and these changes will cause changes in these molecules. How? We will see. So when the ligands, they are bound to the receptors will come close to each other, and they will form dimer. Then there will be autophosphorylation, and this autophosphorylation is detected by these proteins, GRB2 and SOS proteins. And these molecules are going to activate the RAS. How? They will remove the GTP, and GTP is attached. Okay, and again another process is the activation of PLC gamma protein, phospholipase C gamma, and again it will activate the IP3. And as you remember, the IP3 is, I told you that it is involved in the activation of the calcium, because during the growth process calcium is very important during the fertilization. So that's why IP3 is involved, as well as the RAS protein is also involved. So the third process will be, you will have activated RAS protein. What this activated RAS protein will do, this will activate RAF protein. Okay, so this RAF protein is going to activate the MEC enzyme, and this MEC enzyme is going to be phosphorylated, and after that the MAP kinase is activated through the phosphorylation, right? And this activated phosphorylated MAP kinase will go inside the nucleus. Inside the nucleus there are some transcription factors present, and these transcription factors they are also present as inactive form, but when they see phosphorylated MAP kinase, they become activated through the phosphorylation. Okay, and they are going to activate transcription of the response genes. What are those genes? These are the genes responsible for the division of the cell. These are the genes responsible for the growth of the cells. Here you see, and how they are going to be inactivated, that when the GDP is removed and GDP is gained by the RAS protein. So that will be the inactivation of the receptor tyrosine kinases. Okay, so they are also tightly regulated. Here you can see that it is a negative. This is the dotted line is showing that there is a negative feedback. When enough response is generated in the nucleus, it will give a negative feedback to the receptors, and the receptors will be removed to the receptors, and this receptor will no longer will be having the ligands, and they will stop these receptors to get more ligands attached on it, right? So this is negatively feedback mechanisms, and it is very important in the regulation of these receptors, because they are highly involved in the cancers. If you see breast cancers, liver cancers, all types of cancers, you will see the enhanced receptor tyrosine kinases. Okay, so that's why nature has given tight regulation over on this receptor. This is one slide that is showing that when you are going to treatment of the cancers, you just stop the receptors, just block the receptor